Alright, so today we're going to do a little bit on PCR. Originally, if you look on your calendar, I had you doing the lobster. I kind of want you to have a little bit of a basis behind what PCR is before you do the lobster so that you have a little bit of an easier time with the theory. My original idea was for you to do the lobster and kind of play and learn that way, and then we'll discuss it. But what I'll do is making the screencast, listen to it now, you'll do the lobster tomorrow, and then I highly suggest you coming back to this screencast to then go back over everything so that you can make sure again that you fully understand everything, that you fully understand the content that is being described and presented to you. It will be exceedingly apparent who does and who does not. Go back and re-go through this because we will be modeling this next week, and how you model kind of explains a lot about what you get and what you don't get. So let's make sure we listen today, we'll play tomorrow with lobster, and then let's follow up that experience with coming back to the screencast to make sure again that you fully understand everything that is being presented to you about PCR. So PCR is known as polymerase chain reaction. That would be a great thing to put into your notes, polymerase chain reaction. And like I said, when we come back tomorrow and we look at this, this will be where your lobster is situated. And it is under P, but not for PCR, for polymerase chain reaction. So when we first made copies of a gene, this is early on done with E. coli, what we did is we made something called transgenes, where we were able to insert that gene into a plasmid, like what you saw with the bacterial setup and the bacterial explorations within lobster. Plasmids are these ancillary sections of DNA that are usually there to help the bacteria survive in a given environment. Well, there's no given environment with these because they're grown in a lab, so they're grown in optimal conditions. But these genes are cut or spliced into a recombinant plasmid, or they're spliced into a plasmid to form a recombinant plasmid, and then these plasmids are then put back into these bacteria. And once they get put back into the bacteria, then they're just able to grow and divide. So we take a human gene or a eukaryotic gene, we have a eukaryotic cell here, insert through recombination into a donor DNA or inside that plasmid DNA. Once that gets placed into the plasmid, that plasmid then goes into all of these bacterial cells, and then what happens is you have lots of copies of that gene inside each and every single one of these cells. But what problem exists with this? What do we have to do after this? So you want that gene, but what do you have at the end of this process? Is it the gene, or is it more than the gene? So how this is done, so we will go into this more towards the end of the month, beginning of next month. We will use restriction enzymes. Those restriction enzymes will cut the plasmid, will also cut the gene out, so that once it's inserted in, they both have the same sticky ends to make this plasmid, and then you'll make lots and lots of copies of those bacteria. But in truth, it'd be so much easier if we didn't have to use bacteria every single time. So that's what PCR does. So like I told you before, PCR stands for polymerase chain reaction. Again, a wonderful thing that should be in your notes. So polymerase chain reaction is a way for making many specific copies of a segment of DNA. After your 30 rounds of PCR, which is usually how long we'll go through, we'll talk about that in a second, you have up to like 1 billion copies of that specific section of DNA that you are looking for. So you go from 1 to 1 billion very quickly. So it is very efficient. It's very sensitive. It's a high quality photocopy machine. But instead of making copies of dittos or worksheets for you guys in class, it is making copies of DNA. So you only need one cell or one section of DNA to start. Like I said, you just take that one and then you go through this process multiple times, making so many of these copies of DNA. And by the end, you have a large number of that DNA that you are looking for. So you don't need bacteria, you don't need plasmids. You'll see next year, E. coli are wonderfully smelly creatures, so no more smelly friends. That you can just do this all within like a tiny machine. So, PCR process. Just like if I went upstairs to make a copy of a worksheet that I wanted to provide to you guys, there would be certain things that the photocopy machine would need. Well, one, I would need the photocopier, but then I would need all of the toner and the parts and pieces inside of the machine to actually function. I would need the toner or the ink to put that image, put that data onto the piece of paper, and then at the end I'm going to have many copies of the worksheet that I'm trying to provide to you guys. It's mostly the same thing that's going to start here. So you're going to be copying DNA in these microcentrifuge tubes. Now these microcentrifuge tubes, there's all different sizes. We are going to use for PCRs called a 0.2 microliter. So it is a very small PCR tube. It is treated with enzymes in it to make sure that it does not break the DNA or it does not denature it, that there are no ions or anything else inside of it that would harm that sample of DNA. So what are some things that you're going to need? You're going to need that template strand of DNA. You're going to need your DNA polymerase enzyme. We'll talk about this very soon. This is not the same DNA polymerase that is inside of your cells. This is a special DNA polymerase because of the temperature that we are working at. You're going to need your nucleotides. So if you're making copies of DNA, you're going to need nucleotides to actually do this. It's kind of like I need the toner to be able to make copies of a worksheet upstairs at the printer. The other thing that you're going to need are you're going to need primers. So the same way that DNA polymerase in a replication frame of reference, you need a DNA polymerase to be able to start off you needed a primer to start your DNA polymerase off. Well, here you also need a primer. The one difference is in DNA replication you use an RNA primer. In PCR you are going to use a DNA primer to start this. So there's going to be no need for pol 1 to go back and remove it and insert in new nucleotide sequences to make up for those RNA like this happens within DNA replication. So here it's a DNA primer. It can completely be incorporated and elongated from that primer. The nucleotides also, like you can see right here, they're coming in as triphosphates like we learned before. They come in as a triphosphate, lose 2 phosphates, and that reduction in phosphates is the energy, the chemical energy needed to elongate out the strand. So primers are very critical. So we need to know a little bit about that target sequence that we're looking to make copies of, because the primers are a specific region within this sequence of DNA. You want it at almost like the bookend. So I want it at the beginning of the sequence and the end of the sequence, because my idea is you want to copy in towards each other. So you want the forward and reverse primer to be working, so you are actually making lots and lots of copies of what is in between those 2 bookends, what is in between those 2 primers. So for this, we need to know what that sequence is to design the primers properly. Because if we are trying to look for sickle cell anemia, that is one set of primers. But if we are looking to, like we've seen them be used recently with SARS-CoV-2, if we are looking for that, that needs to be a specific primer for that to work. If you remember at the very beginning of the SARS-CoV-2 outbreak, there was a problem with the test that was designed by the CDC and NIH for detection of SARS-CoV-2 at the very beginning, last March. Whoop, lights went out. Well, whatever. So last March there was a problem with that. The reason why this was a problem was because the primer sequence was off. Since the primer sequence was off, then everything was kind of like it didn't work out as well. It wasn't as good, or it wasn't as, there they go again, it wasn't as efficient at finding that specific sequence. If that primer is off, you will not be able to copy in. You will not be able to make enough copies of that target sequence that you are looking at. It needs to be very, very, very specific. And also, when you are taking your primers, you have 6 feet of DNA, but your target sequence is probably small, and in comparison, you're probably looking for something that's a few inches in length. So you want to go from 6 feet to 2 inches of DNA. To do that, you're going to need to cut down and trim, because you honestly don't care about that whole 6 feet of data. You only care about that target sequence. In total, we do this about 30 times. So the process will go from denature, anneal, extension. That is one step, that is one cycle. So what'll happen is it'll go through that, and you'll go and repeat and repeat and repeat multiple different times. So those 3 steps will be repeated for about 30 cycles. It is dependent upon how much of your sample, how many cycles you need. So if you only have one DNA cell, or one cell with that DNA, then you're going to need a lot more cycles. But if you're at a crime scene and there is tons of DNA in your sample, you might need less cycles. So why so many cycles? So look at what happens with the strands. Each time they're going to, this is your full set of DNA, now they get a little bit shorter. Now they get a little bit shorter here. Finally, this size right here, this is the actual piece that you want. And now every single time that you do this, you're going to be making more and more and more copies of just that target region. Not with the other 6 feet of information, just that target that you are looking for. Okay, so with the forensics lens, this also helps so much, because you can go in, like we said, with very little DNA data, and then make these copies, and now all of a sudden go from having a little bit of data to having a lot of data. So your PCR process, what you're going to do is first you're going to denature your DNA. No more need for a helicase. We're just going to jack up the heat to about 90 degrees Celsius. And what this is going to do is it's going to be warm enough to separate the hydrogen bonds between the 2 strands and be able to kind of open them up. Annealing DNA is where the primers are going to attach to the DNA strand. An extension would be the elongation from that primer to the end of the sequence. Okay. So if we were talking about our personal DNA polymerase, so what we use with DNA replication is DNA pol 3, if we were to use that at 90 degrees Celsius, that enzyme would completely denature. So scientists needed to find an enzyme that would not denature at the 90 degrees Celsius. So what they went and they took some bacteria from a bacterial mat at Yosemite, and they were able to find Thermus aquaticus. So this is a bacteria that is an extremophile that is known to live at these high temperatures. So since it naturally occurs at these high temperatures, 90 degrees Celsius is not going to denature that protein. It is still going to be functional. So that is why we use Taq polymerase. Another good thing to write down in your notes, Taq polymerase for our DNA polymerase. So remember, we have 20 to 30 cycles, 3 steps per cycle. Each cycle is about 30 seconds. So again, we'll have our DNA. It will denature here. That denaturing occurs between 94 and 96 Celsius. The low end is 90. That is enough to remove these hydrogen bonds and to unzip that sample up. The annealing is approximately at 68. The annealing temperature is dependent upon what the primers are. It is your lowest temperature, always recognize that. Somewhere in the 50 to 60 range that number is dependent upon which primers you use. So depending upon the primers, depending upon the length of the primers, depending upon the nucleotides present in the primers, will determine what temperature your annealing temperature is. Your elongation is about at 72 Celsius. So that is what we're going to use. That is where thermus aquaticus is most active. So you want to put it at the temperature where your thermus aquaticus is going to be fully functional for it to elongate out. And the more cycles you do, the more of your target sequence you are going to get. So just another image, so you can see high temperature for denaturing, lowest temperature for your primer annealing. Your primer extension will increase for, I said before, for your where thermus aquaticus is going to be most active. It is also at a higher temperature so that the nucleotides are more discriminatory. So the primer annealing is at a low temperature to engage the binding for happening. But once we start and we're looking to extend, we're looking to elongate out with those DNA nucleotides, with those triphosphates. You're going to want it at a higher temperature so that it's harder for the nucleotides to stick together. This will ensure that A is binding with T and G is binding with C. If it was at a lower temperature, it would be a little bit easier for them to mess up. Having that higher temperature makes for a lower affinity. The lower affinity means that it's really going to interrogate. There is no proofreading mechanism with this. So that little bit of higher temperature for the extension process allows for a proofreading mechanism without the addition of an extra enzyme. This is going to be what we're going to be looking at when we model. So we will have a section of DNA. That DNA will get broken apart. It will get denatured. You will have a series of primers that will come in, and then you will extend off from those primers, creating, like you see here in gray, a new series of DNA or a new chain. Once that chain gets fully elongated, you will now go from two parent strands to then 4, from 4 to 8, and from 8 then we'll keep moving and keep cycling, each cycle becoming more and more as your total number. These short PCR products that you see here, these are what your target sequence is. That is the target sequence at the intended length, where you see primer 1, primer 2, and the only piece is the bookends between those two primers. So what we are going to be looking at is we're going to be looking at both Labster and Lab Exchange as ways to explore out what PCR is, make sure that you have a concrete understanding of this. PCR is kind of the basis for, or the preliminary step to get to gel electrophoresis. PCR is the basis for us to be able to do sequencing, which was the basis for us to understand CRISPR. So it's kind of like this really simple process that is an incredible diagnostic tool that enables us to learn so much about our sample and about what is going on. So it is something that is incredibly important, and we need to spend a little bit of time with that. So we'll be doing some Labster, we'll be doing some Lab Exchange, and we will also be doing some modeling with PCR and with gel electro before we move fully into gel electro and restriction enzymes. So again, goal for this, you want to be able to come back into these slides, look through, make sure you are okay with this. These slides are from 50 to 68. So you got about 18 slides in there for you to look through. I would listen to this lecture, take down your notes. Tomorrow we'll go on Labster, we'll do some modeling, we'll do some Lab Exchange. Make sure you keep coming back to this Loom to make sure that you fully understand what PCR is. Again, it is a simplistic process, but it is the basis of so many different medical diagnostic tools. It is the basis of technology for what we are going to be exploring moving forward. So it is incredibly important that you spend some time with PCR and that you really understand what is going on and how it works. All right? Let me know if you have any questions. Shoot me an email, and I will see you all soon.